All right. Cellular respiration is the story of how a cell makes ATP, right? If you remember all the organelles, do you remember where ATP is made?
Mitochondria.
Yeah. So this chapter is sort of like photosynthesis. It's kind of like a reverse process. This chapter is all about breaking up something to make energy, like catabolic reactions. Photosynthesis is going to run the opposite direction. So photosynthesis is about making things, like taking energy and building something. This chapter is about taking something and breaking it up. Food, really, that's what it comes down to, our food. So it's going to be, and so for food here, we're going to use glucose to represent food, because there's three different types of like energy that we get out of our food: carbohydrates and the other two, carbs. What were the other two? Carbohydrates,
Lipids.
Lipids and proteins. Thank you, online people. Carbs, lipids, and proteins. We're going to take carbs because carbohydrates goes through the longest process. So when you understand carbohydrates, then we understand like everything else. So for carbohydrates, we're going to use glucose. So, I mean, that is kind of one of the main things that our body uses for energy. We're talking about blood sugar. So our cells are going to take in this blood sugar. What are we going to do to it? Well, we need something else besides glucose. We need oxygen. Glucose plus the oxygen from breathing in. And that's going to we're going to use that to put the ADP and the P back together. And then there's a waste product. You breathe in oxygen, you're going to breathe out carbon dioxide. And also water. Water is a waste. Is not a waste. It's a byproduct. Water is a byproduct of this. So that's that's almost the formula. I'm just going to take glucose. I'm going to rewrite it below. So for glucose, technically, and this is kind of one you should know, it's C6H12O6. That might be one you'll see again. But we're going to kind of abbreviate it here. I'm going to just put C1H2O1. But, and there's ones here, but I don't need to put them. So I just put CH2O. So that's a shortened form of a carbohydrate. In fact, sometimes they'll just put CHO, and that just means carbohydrate. But technically, it's C6H12O6. So I'm going to rewrite it. CH2O plus O2. And then on the other side of the equation, I'm going to keep the ATP out for a second. CO2 plus H2O. These are the things that go in. These are the things that come out. Glucose and oxygen go into the process. Carbon dioxide and water come out of the process. And if you look, it's balanced. Like like there's the C. I take the C and I put it over here. So like if I move that carbon over here, then I've got CO2 and I've got the H2O here. So it's just, so from sugar and oxygen, we get carbon dioxide and water. And of course, ATP is made. I just didn't put it in here. Photosynthesis goes the opposite direction. Plants are, and that's going to be Wednesday. We'll talk about it. Plants are going to take carbon dioxide out of the air and water from the ground, mix it all up, and make sugar. And they're going to release oxygen gas. But right now, cellular respiration, it goes the opposite way. We're going to take sugar and break it up. We're going to take oxygen gas, mix it all together, and we're going to end up with ATP, but we're going to end up with these two products. Carbon dioxide, which we're going to breathe out, and water, which we can use. There's going to be 3 steps to this whole process, and I made it as short as I can. Like I took out all the extra steps that are only going to inhibit you from understanding. It's important you understand like what's going on here. This is just showing you what I'd written before. See, look what we're skipping. Oh. This is reminding me to tell you something. You were going to see 2 molecules when we talk about this chapter and photosynthesis. You're going to see 2 molecules, and they either start with NAD or FAD. And there's different versions of them. For example, if you look up here, it says NAD and it's positively charged. You might not be able to see that. This one says NADH. It doesn't matter. If it says NAD or FAD, these are molecules that carry electrons. So NAD and FAD are electron carriers. They're carrying electrons around this process, picking them up in one place, taking them to another place. These are actually vitamins, NAD and FAD. So this is NAD, this is just FYI. NAD is niacin, and FAD is riboflavin. So a lot of these B vitamins, like niacin, riboflavin, pantothenic acid, some other ones, biotin, ones that I don't want to try to recall. But a lot of the B vitamins, this is what they do. They don't actually give you energy, but they help cellular respiration. You need them to convert the food you eat into ATP, because that's the goal of this lecture today. We want to convert the food we eat into ATP, because that food is potential energy. We want to make it free. We want to free that energy up so we can use it. Sorry, guys. NAD, FAD, don't worry about the form that it comes in, because I never talked to you guys about oxidation and reduction reactions. So it's a little too late to, wrong one. It's a little too late to start now. Okay, so there's 3 steps that I'm going to talk about. One's here. Step 1 is going to be glycolysis. Step 2 is going to be Krebs cycle. And step 3, we are just going to call it the electron transport chain. I do have a step 1.5, so just So glycolysis, Krebs cycle. Krebs cycle is sometimes called the citric acid cycle, depending on what book you read. Krebs cycle is kind of an older name. So new books will call it citric acid cycle. Or even TCA cycle. I'm not in favor of making words longer, so I just stick with Krebs cycle. And then electron transport chain, we're gonna group it together with this thing called oxidative phosphorylation. We're gonna like kind of ignore that. We're just gonna call the whole thing electron transport chain. So you'll notice that glycolysis is happening out in the cytosol. I don't know if you guys remember what the cytosol is, but it is the liquid. It's the liquid that everything's floating in. And sometimes we call it cytoplasm, but like technically speaking, cytosol is the liquid. And then the other two steps, Krebs cycle and electron transport chain, those happen in the mitochondria because we know that that's where ATP is made. So we're going to talk about each of these steps. So we want to convert food into ATP. That's our ultimate goal. And the food that we're breaking up is glucose. That's what we're using. That's what we're talking about, glucose. How do we take glucose in our blood sugar? How do we convert that into ATP that I can use to make my muscles work? Right. So the first thing, there's two problems with molecules. One, they're too big. So when you have glucose in your blood, it's still too big. It's too big to get into your cell and process. We have to like cut it down and trim it down and make it smaller. Right. Starch was too big to get into your blood. We had to break starch up into glucose. And glucose was small enough to get in your blood. Okay, now it's in your blood. We got to get it into the cell. Once you get it into the cell, it's too big. We can't fit it into the mitochondria. If we follow, you should have an idea about like how big some molecules are. And when we follow molecules, we use carbon. In biology, we talk about carbon. This is a 6-carbon molecule. This is a 5-carbon molecule, whatever. Glucose is a 6-carbon molecule. That's just to give it a size in comparison with something else. Like I'll give you pyruvate is a 3-carbon molecule. So you can kind of see what's going to happen. We're going to turn one 6-carbon molecule into 2 3-carbon molecules. So we're just taking a molecule and we're just breaking it in half. That's essentially all that's going on here. So we got to start making this molecule smaller. It's physically too big to get into the mitochondria. In fact, the mitochondria doesn't even like things with 3. This is a 3-carbon molecule. It's actually a little bit too big still. We're going to have to trim it down. But anyway, glycolysis is 1, making it smaller. So we got to make this thing smaller. 2, we got to change things around a little bit. Like the carbons are going to stay where they are, but we got to move the oxygens and the hydrogens around. For some reason, you got to like do it a little bit different. Be like changing the clothes. You got to change clothes to go to the certain place. Can't show up to the mitochondria in business casual. You have to, it's black tie. So you got to switch some things around. All right, so let's go into glycolysis. Help if I erase these. Yeah. Here's half of it. A lot of places would make you memorize every step, and I used to as well. So you would have to know that like glucose goes to glucose 6 phosphate, and you would have to know that like hexokinase was the enzyme that catalyzed that reaction. And then what happened? Students were doing just what I would do. You're literally memorizing everything, and then I would say, "What's the purpose of eating food?" And you'd have no idea. I don't know. Glucose 6 phosphate, catalyzed by hexokinase. I'm like, yeah, but what's the point of eating food? Why are you breathing air? What's the point? No idea. So I was like, well, do you really need like these little things? No. If you get stuck taking organic chemistry, then yes, that sucks to be you. That's a different class. So I took it out and I found that people did better. I just want you to get what's going on here. So we're going to not do this. I want you to try just to, it's not easy, but try just to look and see what's going on. This thing looks like a hexagon. Is that what you call it with 6 hexagon? Every time it bends, that's a carbon. So it's like 1, 2, 3, 4, 5, 6 carbons, right? 6 carbons. Now it looks a little bit different. It looks like a pentagon. 5 carbons. There's another pentagon. So it's 5 carbons. And then finally, when you get to the end of this slide, it's 3 carbons. 1, 2, 3. So there's actually 2 of them. There's 2 of these 3 carbon molecules. So to summarize all of this one molecule of glucose broke in half. You started with, I'll use like these circles to represent glucose. You just went like this. That's it. You're breaking up a molecule. What happens when you break up? Well, let me not ask that yet. Let me go to the next slide. All right, I'm going to continue. This molecule here, glyceraldehyde 3-phosphate, you do have to know it, but we're going to abbreviate it. We're going to abbreviate that molecule G3P. You will see maybe that molecule's name in the future, like maybe in a future class. When we talk about physiology, we talk about G3P. Just know in your head, that's got something to do with like breaking down my blood sugar. When I break down my blood sugar, I get this glyceraldehyde 3-phosphate, this G3P. Just kind of remember in your head when you see it. Actually, we'll also see it in the chapter on photosynthesis. Oh, check it. Right to it. This is the other half of it. Like it continues. It continues. 3 carbon, 3, 3, 3, 3. It stays as 3 carbons. Like the size of the molecule doesn't change from here to here. From when we're at G3P until we get to pyruvate. Pyruvate's the end. It's still 3 carbons. So, like, if you look at what's happening, we're just switching things around. We're switching around hydrogens. We're switching around oxygens. We're trying to like change the shape of it to make it something that will fit into the Krebs cycle. And you see like these, you see some things in yellow here. One thing you see in yellow is that it says NADH. That's NAD. NAD is an electron carrier. It's carrying electrons from that piece of G3P. It's carrying the electrons somewhere. We'll get to that later. And then you see some ATP here. That means like we're getting some energy from all of this. My question is, why maybe it's a difficult question. Why are we getting energy from breaking down glucose? Like, what do you know about energy?
Is it a canatonic reaction?
Yes, almost. Except you said the word that I always put as well. Catabolic.
Catabolic.
But I knew you knew it. Right, exactly. It's catabolic reaction. You're breaking something down. When you break something down, you release some energy. So we do get some ATP from this, but not a big deal. Right? So let me go back a little bit. Let me use this slide. So what are we getting from glycolysis? Like, what's it all boil down to? We're getting two things. One, we're getting 2 molecules of pyruvate. So we're taking 1 glucose and we're making them 2 half glucoses, right? So 2 molecules of pyruvate. And we're getting a little bit of energy from this, just because of the fact that it's a catabolic reaction. We're breaking down a molecule. When you break down a molecule, you get some energy. This is not our big energy payoff, though. That doesn't come till the end. Big payoff of energy is here. This is lagniappe. It's bonus ATP. We'll take it. It's not the reason that we're doing it. We're doing it to get pyruvate because that's like an easier molecule. Because eventually we want to get pyruvate into here. So we have to tweak it a little bit. So we're getting 2 molecules of pyruvate. We're getting 2 ATP like profit. It's like you put in 2 as an investment and you get back 4. So anyway, profit, we get like 2 ATP. That's it with glycolysis. We're turning glucose into 2 molecules of pyruvate, and we're getting a tiny bit of ATP out of this. And that's glycolysis. Without all the molecules. Because you'll forget the molecules. It's like I forgot most of them. Everybody forgets them. Go to a doctor and just say, "Oh, can you tell me the steps of glycolysis?" They'll be like, "No, I don't remember that." So between step 1 and 2, we've got this step in the middle. It's going to go right here. It's like a step 1.5 because we have to tweak pyruvate a little bit more. Pyruvate is still a little bit too big. Pyruvate is 3 carbons big. We have to make it 2. We got to trim off one of those carbons. And we got to do something else to it. We have to take this thing called a coenzyme. Coenzymes help enzymes. So we have to take this coenzyme and you'll see. Oh, this again. There. Okay. So we're going to tweak this molecule a little bit. Here's our pyruvate. There's pyruvate. 1, 2, 3 carbons. We're going to take one of the carbons off. In fact, we're going to remove this whole thing right here. Not that that's super important, but this is, you can see it, carbon dioxide. So we're going to take that off. We're going to throw that out into the bloodstream. That's going to make its way up to your lungs. You're going to breathe it out. So this also answers one of the questions. Why do you breathe? You breathe. People's first answer would be, I breathe to get in oxygen. That's like a partly true answer. You're not actually breathing to get in oxygen. What's making your body breathe is getting rid of carbon. So you get rid of carbon in the form of carbon dioxide. So it's like, if you look at oxygen, this is like what you're breathing in, and then this is what you're breathing out. So the difference between those two molecules is carbon. So it's like the oxygen's going in, grabbing a carbon, and then going back out. Right? So you're trying to get rid of carbon in your body. There's different ways to get rid of carbon. You can poo it out, you can pee it out, but what about this stuff that we're actually using, right? Your cells are using some of this stuff to make energy. So we got to get rid of that a different way, right? Your cell's already taken it in. It's too late. It's too late to make that into like feces. It's too late now. The cell took it in. Like it's in our blood. Like that's it. So we got to breathe it out. We don't need all this carbon. It builds up in us. It's acidic. Carbon dioxide's acidic. So when you die from like asphyxiation, it's not that you didn't have enough oxygen. It's that carbon dioxide built up too much in your blood because you couldn't get rid of it. That made your blood acidic. And then that does stuff, right? It messes up your enzymes and it'll start breaking open your cells, and that's it, you're done. So breathing out is really getting rid of carbon dioxide. That's what's driving expiration, like breathing. That's why you breathe. To be like really technical about it. But if you want like a little bit of an easier answer, why are you breathing? Why do we breathe? So we got to get rid of that extra carbon from our food. That's it. That's the answer. That's why we do it.
Now that you mentioned that, it's like I'm an asthmatic and I get asthma attacks, and sometimes when I get them, the biggest problem is that it's not that I can't breathe in, it's that I cannot breathe out. And that happens.
It's making your brain, like if you ever were to like panic or something, that's what's making you panic. The buildup of carbon dioxide. Your body's not, oh shit, I'm not getting enough oxygen. It's like, no, this dude's got to get rid of carbon dioxide. It's too much building up. Why is he not expelling it fast enough? So it's making your little bronchioles constrict, asthma. Yeah, it sucks. So we're going to take this, we're going to remove this part up here, we're going to keep this part down here, and we're going to add something called coenzyme A to it. And so we get this molecule right here called acetyl-CoA. CoA stands for coenzyme A. I don't care that you know about coenzyme A. This is one of your B vitamins, by the way. What I care about is that acetyl-CoA is the thing that's going to go into the Krebs cycle. We've tweaked pyruvate. It's only 2 carbons long now. We got rid of one of the carbons. So it's only 2 carbons long. Wait till they pass. They're here on the computer. Hey, how's it going? Hey, how's it going? Hello. Hi, how you doing? Hey, morning. How you guys doing? Hey, how's it going? Hey, hi. Something going on. The important thing is I got out of it. Thanks to you guys. Oh, she was saying the small class sizes were like something positive. There's a sweet spot for class sizes. It's right around 10 to 15. If you get like below 10, it starts to fall apart for some reason. You think that if it was like 3 people, then we would have like a great class. It doesn't work. And then, when you get above 15, it starts to go down. If I only had 3 of you, we wouldn't even have it here. We'd go to the cafe. And we'd go sit down at a table. Do you guys get what's happening here? There's a lot of stuff up here that I'm kind of, we're taking pyruvate. We've got to rip off a carbon and convert it into a molecule called acetyl-CoA. I'm going to go back again to that one photo that I have. And I'm going to add that part in. Right here. So we're going to convert pyruvate to that molecule called acetyl-CoA, coenzyme A. That is what is going to go into the Krebs cycle. Now this is small enough, and we've tweaked it enough, and we put that coenzyme A with it. It's got to be with it. I don't remember why, but it's got to be with it. Now we can put it into here. So that's kind of the step 1.5.
Just
Didn't
carry.
go away, did it?
No, just carry through the wall.
It's going to say goodbye to everyone.
That would be equally as hilarious.
Let's shake each one's hand. All right. So pyruvate to acetyl-CoA. Acetyl-CoA is going to go into the Krebs cycle. So let's talk about the Krebs cycle. The Krebs cycle is a shredding machine for molecules. It's a shredder. We've got these 2 molecules of acetyl-CoA. They've got like 2 carbons each. We're going to shred it up into nothing. By the time the Krebs cycle is done with it, it's nothing. Acetyl-CoA is something. Acetyl-CoA used to be pyruvate. Pyruvate used to be glucose, which is your blood sugar. Glucose used to be, let's say, starch when it was in your stomach. Right? And that starch used to be a biscuit. So you ate the biscuit, broke it down into you liquefied it into liquid starch, and then you broke it into glucose, and your blood sugar went up, and some of that blood sugar went inside of your cells, and your cells broke it in half, and then tweaked it into acetyl-CoA. This is the last stop for your biscuit. After this, there's no more biscuit. I should stop saying biscuits and go to the slide.
You said it was a molecule shredder?
Yes.
Molecule.
That's what I'm calling it. It's actually, I'm going to say 2 things about it. It's a molecule shredder and an electron harvester. Because that's the real purpose of this Krebs cycle. Not to shred the molecule. So what? We don't care. We want the electrons off of it. Your food has electricity in it. We want to pluck all that electricity out of your food. We're going to use it. We're going to use it to make ATP. So there's energy in, there's the acetyl-CoA. This Krebs cycle keeps going around like this. And it's hard to see, but if we were to follow these, like look at oxaloacetate, it's 4 carbons long. So I'll just put like a little 4 right here. 4. Anyway, it's 4 carbons. It's coming along. It's picking this acetyl-CoA up. It's grabbing it. Acetyl-CoA is 2 carbons. So the 4 carbon oxaloacetate, you don't have to know the names, but the 4 carbon oxaloacetate picks up the acetyl-CoA And now, here it is, it's 6 carbons long. And then it goes through citrate, isocitrate, keto. And so 6 carbons. One of them's got ripped off. Now it's 5 carbons. Another one got ripped off. Now it's back to 4. And each time one of those carbons gets ripped off, you see right here, carbon dioxide. That's going back out into your blood, lungs. You're going to breathe it out. So this was the last, this is the last, the end point of your, what? Biscuit. This is it. Oxaloacetate comes by, picks it up, rips one of the carbons, throws it out, rips the other carbon, takes the other carbon, throws it out. It's nothing. It's no more molecule. Just a bunch of atoms. Where do the carbons and oxygens go? They went out. You breathed it out. There's some hydrogens there. We'll keep those. We can do some stuff with them. But really, we wanted the electrons. The Krebs cycle wants the electrons. Pull the electrons off. You see NAD, NAD, FAD, NAD. There's all these electron carriers. They're grabbing all the electrons. They're going to carry them over to the last phase. So we're collecting a bunch of electrons. That's what the Krebs cycle does. You don't have to know all these citrate, isos, whatever it is, ketoglutarate. That doesn't matter. What's happening is that you get the idea. This happens in the mitochondria. We're shredding acetyl-CoA, what was left of our glucose molecule, and we're pulling electrons off of it. And it just keeps going around. It goes around, it grabs another acetyl-CoA, rips it apart, comes around, picks up another one, rips it apart. It's just constantly doing that. And all these NADH molecules are grabbing the electrons and carrying them, taking them away to another place. We do get some energy from this. But again, it's not like a huge payoff. Why does it do that? All right. So Krebs cycle, what did we want to get out of the Krebs cycle? Harvest electrons. I'm just going to put that for electrons. We want to get electrons. We do get some ATP because, again, this is a catabolic reaction. We get another 2 ATP. Nothing to write home about. But you want to harvest electrons. You want to get a couple of ATP. Not a big deal. Big things are the electrons. I'd fall asleep if I was sitting down the whole class. I put myself to sleep. And that's how boring it is. It's like, oh, who's talking? Why won't that guy shut up? Oh, it's you, you're talking. Well, why won't you shut up? Because you get paid for this. You gotta keep talking. Any of you want to talk? Do you have any questions? What time do you guys have?
9:46.
Oh, we've only been talking for 45 minutes? Can you believe it? Haven't you felt like you've been here for like an hour and a half?
I was really hoping.
That'd be it. wondering if we should just take a break.
Or,
Yeah.
or we can go until 10 o'clock, take a break then. So
Yeah.
when we come back, it might go, it feels like it goes a little faster.
I always hated when I worked like shitty jobs. I worked a lot of shitty jobs like you guys. The worst thing is when like the manager would come after like 3 hours. Okay, go to lunch. Oh, kidding? I gotta go to lunch, and then I got like 5 hours left when I get back. I hated that. I always try to take my lunch like 5.5 hours into the shift. Then you get back from lunch and 2.5 hours, and I got a 10-minute break in there. That I could do. Oh, thank God I don't have lunches and breaks anymore. I can never go back to that life. Wow. If you're looking forward to your 10-minute break or 15-minute break, you need to get out. Get out of your job. If you're counting down the time to your break, get out.
We give you 30 minutes now.
For lunch or for a break?
Whatever. For me, it was for lunch. You ain't never had a break. Just lunch break was your break.
Yeah, I don't. If you work at a place like Walmart, they make you, like, you do 2 hours, so you get like a 10 or 15-minute break. They pay for that. And then you got to clock out for your own lunch. But some people, like, I don't know if you ever heard, like if you're at Walmart and you'll hear the cashier, oh, I'm about to go on break. You mean like lunch or like your 10-minute break? I'd rather work through that. Because I just can't stand, it's like, as soon as I go on break, I'm going to have to come back. I'd rather just work through it. Nothing you could do when you're at Walmart. I feel so bad for those cashiers. Just putting things over the scanner. If you work faster, you just get more customers. There's just like nothing you can do. You're there. You're there until it's time to get off. And then you get home, and then you start thinking about how you got to go back to work the next day. So that day's ruined. You can't watch The Bachelor or whatever people watch. Talking crap. All right. Breaking down sugar. Harvesting the electrons from the rest of that molecule. And we're going to take all those electrons and we're going to take them over to the last phase, which is called the electron transport chain. When we get into the electron transport chain, there's some ideas that we're going to talk about from like the last chapter or a couple chapters ago. One is active transport and passive transport. Like moving through a membrane passively, like diffusion, and actively we have to pump it. So the mitochondria is going to do both. So if you look like at this mitochondria, you see how it's got like these things coming into it. It's kind of like weird shape. Well, why don't I just show you the real one? You guys can't see yet. Oh, hey, look at that. I like that photo. Hold on. Before I jump into it. Why is it on that one? That's just like another, that's the same thing, which I probably should have put it on this. It's doing like the circles like I was doing on the side there. Pyruvate, we're going to shave one of the carbons off and turn it into acetyl-CoA. Acetyl-CoA enters this Krebs cycle. This gets ripped apart. And we take the electrons off of it. We do get a little bit of ATP, but it's really the electrons. And then where are we taking all those electrons? Not there, not there. We're going to talk about this. So this is the mitochondria. You see how it's like, it's got like a dark brown and it's got a brown and a yellow color to it. So it's got like these two membranes. Like this is a membrane here. That's one membrane. We don't really care about that one. But this is a second membrane inside. like those lines. I'll do another one. So it's like a double membrane thing, right? So that's what we're talking about here. That's this, where I'm pointing to with the arrows, the inner membrane. The mitochondria is going to do stuff with that inner membrane. So in that black, see these black lines right here? All these black lines, that's this. It's a phospholipid bilayer. This is the layer, this is just the same thing that surrounds our cells. Like what's the wall? Like we don't have cell walls like plants do, but instead we have a phospholipid bilayer. 2 layers of phospholipids. So that's a layer of phospholipids, and then that's a layer of phospholipids. 2, right? And so that same membrane exists inside the mitochondria. They think that the mitochondria was, well, who cares, right? Well, I will tell you something else interesting about mitochondria. It turns out it has DNA in it, and it persists longer than the DNA that's inside of our nucleus, right? And the mitochondrial DNA, that's pretty new. You know, like DNA, like, of course, your grandparents' generation, they probably never knew about DNA. Like it existed, like when they were kids in school, it didn't come out until like maybe the 50s. So maybe they just learned about it, right? So it's still like kind of new when you think about science. It's our grandparents. I don't know how old, but anyway, your grandparents' generation, right? And so up until even when I was in college, we didn't learn about mitochondrial DNA. Like DNA was in the nucleus, like with mitosis, right? That's where the DNA is. There's no other DNA. Maury Povich didn't have his thing on TV. So we didn't really know about DNA. They're always the father. That's a show where nobody wins. I'm like, if he's a father or if he's not the father, it really doesn't matter. He's off the air, isn't he? It's been a while. Is he still on? Any of you stay home and none of you watch? See, that's because of Netflix and Amazon Prime. You guys are binging shows because you can't be bothered to watch commercials, and you're missing the classics like Maury Povich. You're missing all the drama hours in the morning. What are you going to do if you need a lawyer? You don't even know who to call because you're not watching TV. All right. There was some point somewhere in here. Oh, the mitochondria's got DNA. That's like a really new thing. It's like a really exciting thing. And a lot of people in prison were able to get out of prison because they could we don't have any DNA from this sample. Yeah, because all the DNA in the nucleus, that kind of degraded. It like fell out and couldn't use it, right? But then they realized that there was other DNA in the mitochondria. We just have to treat this mitochondria a little bit different, and we can get in there and tap that DNA, and it's still good. It lasts for a lot longer. So they go back in, and they could find out new things. Oh, sorry, we put you in prison. Yeah, we heard all the witnesses that you totally were in another state, but, someone had to go to prison. Sorry, our bad. Yeah, so it's an interesting organelle. It's got like some DNA in it. We think that it might have been a living organism at one time, or like that. All right, I'll stop talking about it. What time do you guys have? Let's come back at 10 to 10:15. I can already see you're out. A couple people listening, most of you are out. So I'll when we get back from break, because I don't want to do more than this chapter today. I think it's like it could be it's like heavy material, and I'm going through it like oh, don't you get it? Don't you get it? But there's a lot of crap on there. So let's take a break. Come back around. We'll start again quarter after. We'll take a break. We'll start again at quarter after. I'll go over the first two steps really quick, and then I'll go into this last step because I don't want to do more than this chapter today. Right? Oh, and then we also have to talk about your exam on Wednesday. No, I'll keep this all on. What happens if I stop recording and then start again? I don't know.
Just leave it on.
You guys are good. I think I'm gonna leave the call, and then at 10:15, no, because that'll stop. Anyway, you guys are fine. Just do your thing.
Can you just leave it on?
Yeah, that's what I'm gonna do. I'm gonna stop recording and then start again.
Okay.